And today we have three speakers actually from the BASE2 project, which is the Berlin Aging Study. First it is Denis Gerstorf, who's a psychology professor at Humboldt University and a scholar of lifespan development. And all of the three speakers are co-speakers of the study. And Professor Gerstorf, together with Johanna Drewelies, who is a postdoc, both at the HU, but also at the Gender and Medicine Department at Charité, they have an interest in lifespan development, for example, of personal traits and social activity. While the third speaker, Ilja Demuth, is professor at Charité at the endocrinology department. He's a human geneticist, and he's leading the subsection of the BASE2 study, which deals with geriatrics. So at that, I would like again to thank the three speakers. I'm happy that you're here, and I would like to hand over to you.
Yeah. Okay, can you see the presentation?
Yes.
Okay, so great. Thank you very much for your introduction and also for the opportunity to present the Berlin Aging Study 2, the BASE2 study here in this forum. The Berlin Aging Study was launched in 2009 as a multidisciplinary study, but also as a multi-institutional study. And this is illustrated by those slides here. So starting on this slide on the left, the Charité is representing the internal medicine and the geriatrics part of the study. Then here we have the Max Planck Institute for Human Development and the Humboldt University representing the psychological part of the study. And on the right here, the socioeconomic panel located at the German Institute of Economics representing socioeconomics and the survey methodology. We have two study sites in Tübingen and University of Tübingen and at the Charité for the immunology part, and then at the University of Lübeck we have the genetics and also the epigenetics part of the study. So I'm going to talk a little bit in general on the study, and then I will also focus a little bit on the data represented by the Charité, the University of Lübeck, and also the immunology data from Tübingen and Charité. So as I said, the study was launched in 2009, and one of the main aims was, and it is still is, to identify and to characterize the factors associated with healthy versus unhealthy aging. We have recruited two age groups of participants. The main group representing the older adults aged 60 years and older at baseline. And then we have recruited a group of younger adults as a control group, which were aged between 20 and 40 years at baseline. We have approximately 50% men and women, and all participants were community dwelling at baseline, and they were from the Berlin metropolitan area, which is illustrated by the Berlin city map here on the right. Each dot represents the place of residence of one participant. So as you can see, most of them are resident within Berlin, and a few live very close to Berlin. The baseline data collection took place between 2009 and 2014. So with respect to data assessment at baseline, here at the Charité, we had 2 study days, which were usually 7 days apart from each other. On day one, physicians undertook a comprehensive physical examination and anamnesis, which also included medical and family history. Of course, lifestyle factors were assessed, such as nutrition, alcohol use, and drug use, sleep, and many others. Physical assessments were performed at both study days. The physical status was assessed, functional capacity. For example, we did a comprehensive geriatric assessment, including the TINETI test, timed up and go test, the Barthel index, activities of daily living, and also instrumental tests such as pulse wave velocity, electrocardiography, audiometry. We had DEXA scans and many others. And of course, we also took biosamples. So those were taken on the second study day, blood and urine samples, which were used to immediately assess or determine about 100 laboratory parameters. And some of them are listed in this slide. I'm certainly not going through those, but as you can see, we have already many laboratory values. We used the biosamples also to biobank them for future analysis. And then we already determined various immunological data and also genomic data. And this is shown here. As I said, we started this study in 2009. The genotyping chip at that time was Affymetrix 6.0, and we used this one to genotype almost all of the participants, so more than 2,000. And then later, with the development of the global screening array from Illumina, we genotyped an addition 1,000 of those samples again. And in addition to that, we assessed the methylation profiles of more than 1,000 participants using the Infinium methylation EPIC array. Today, we must say the array number 1, because number 2 is out, as you probably know. Since we are interested in biomarkers, one of the biomarkers we assessed is the relative leukocyte telomere length using a quantitative PCR assay. And one thing which I'm especially proud of is that we were able to establish more than 450 lymphoblastoid cell lines. So those are cell lines established from B lymphocytes by Epstein-Barr virus transformation. And those cell lines had been expanded and then frozen in liquid nitrogen. So those cell lines could be used and have already been used for cell biology experiments, but they could also serve as a source of biomolecules, for example, such as RNA or DNA. And then we performed a number of different tests to test for pathological decline in cognitive performance. And those tests were, for example, the CERAT test plus test battery, but also other tests like the millimeter state examination. We did a clock completion test, DEMTEC test, and also tests to look for depressive symptoms such as the GDS and the CESD matrix. And with this, I would like to hand over to Denis Gerstorf. And for this, I guess I have to stop here.
Thank you, Ilja. So I would like to go on and noting that we not only have a very comprehensive assessment battery of medical testing, but also a very comprehensive psychometric cognitive assessment battery that is shown on this slide. So we have a whole variety of basically IQ tests with some, let's say, 4.5 hours or so of cognitive performance across basically all of the major abilities, including episodic memory, a number of different tests, working memory, a number of different tests, fluid intelligence and reasoning, a number of different tests, as well as perceptual speed, executive functioning, etc. We also have a pretty comprehensive assessment of psychosocial measures, as is shown on this slide here. Well-being, affect, social integration, stress, personality, motivation, and control, as well as the perception of time and people's age. And as you can tell from the distribution here, each of these constructs is measured by a number of different items, if not a number of different scales. And what is shown here in the in brackets are the number of items per scale. This is basically our cross-sectional data. As Ilja Demuth was referring to, it took us about 5 years between 2009 and 2014 to collect all of this, given the breadth of our assessment. And then we moved into extending this cross-sectional data into a longitudinal study. About 7 years later, on average, we have been able to collect our second major wave of data. Many of the constructs have been reassessed, and I guess that you can tell from the numbers here, it's a little different. So let's put it differently. Probably we started this second wave of assessment in, I think it was June 2018, and we were basically done with our older participants, I think in February or so 2020. So right in time before Corona hit us, and we had only started collecting data from our young participants when basically the shutdown hit us. That's why we had to stop data collection. That's why we only have had 64 of the young participants. We had wanted back then to test more of those. In the meantime, we have also collected a third major wave of data. Again, the numbers are a little different. There are a number of different reasons for that. So the let's say smaller number here, like 485 for the older adults, this refers to the medical assessment. The larger number of 825 refers to the cognitive and psychosocial assessments. Probably let me put these numbers a little in perspective. This study was actually part of a separate study that was conducted at the Charité, the so-called Charité Corona cross study that was focusing on immunological questions, and we had informed our base participants about this study and had asked them that they could join this, and about 500 or so selected themselves in. What I would also like to convey are two more things. First one is, this is highlighted by these two check marks here in between. So we have a minimum of 3 data points for many of the major constructs in the medical and cognitive realm. But in addition, we have several smaller sets of data collections be carried out by running some online questionnaires, by running some questionnaires that we had sent them via snail mail. We have conducted some site studies. So for a number of different measures, particularly in the psychosocial realm, we have more than those three data points. And in addition, we are currently in the field again, if you wish. We also have brain scans of our participants, and we are currently collecting the third wave for the subset of, I think it's some 330 or so participants who have undergone these fMRI scans. We are also hitting the field of another daily life study. We have conducted one of those, or the first one, in 2021, so during Corona times, and we will hit the field again in early 2025 to reassess our participants, so as to see to what extent their daily life has changed over these past 4 years. And we are currently also putting together another application to the German Research Council to fund another full wave, and there we anticipate that we will probably arrive at sample numbers of 600 older adults and some 200 younger adults. And with this, I would like to hand over to Johanna to tell you more about how the data can be accessed.
So I'll talk a little bit about the practicalities of the data. Dennis, if you could move one slide. Okay, so yeah, so these are all the steering committee members. I think on these slides, you will also find a link to the BASE webpage, or you've seen it already. And if you go to the next one, on this page, you will also find all the publications that have been using this data, which could be quite helpful if you're considering using the data or you want to see if the research question in mind is already published. You can do that there. If you go to the next slide. And here you will also find a documentation of how you can access the data. I'll walk you through, but it is also described on this page, and all the different documents that you will need in order to apply for the data are also there, including the contact information with our study coordinator. If you go to the next slide. Okay, so I'll do a step-by-step how you can access the data. It looks much more complicated than it is in reality. It's quite simple. So the first, of course, is you need to find your research question and see if the data kind of fits what you're looking for, and discuss this interest with the BASE team. Again, all the information can be found under the link. You can also always contact any of the BASE members directly. And then it is necessary to prepare an application in English because our team is kind of international. And here, the first step is to provide a summary of the data, which is basically a regular abstract of what you would be planning to do, a more or less detailed work plan, and a timeline. So one thing that might be to note is that we say that projects are usually for 2 years, and the reason for that is that we don't want to plan for 10 years, and then we do not know what the data is used for anymore. So we're thinking of kind of a paper-based, so publication-based projects. So a detailed work plan as far as this is possible without having the data, of course, and a more or less precise timeline, and then also a list of the variables that you will need. As you heard before, BASE is a large study with a huge number of variables. Alone looking at well-being, we have over 10 different indicators of well-being. So it is necessary for us, in order to prepare the data, to kind of know what you want to look at. And then most important step is a data protection plan. So you need to develop a data protection plan that details how the data will be handled. And this plan also needs to be signed by your institution's data protection officer. And it might also be important that each data user must additionally sign the data protection agreement, depending on who is going to ask for the data. And then after this application is approved, you will sign a contract of data usage with BASE2. And then the data is provided via secure mechanisms, of course. And then once you have finished the project, there are a couple of steps which are also described in great detail on our webpage, which is basically just citing the study correctly. So nothing that is new. If you go to the next slide. Yes, so we have prepared a couple of Q&As. Of course, you can ask any other questions that might come up later, but these were the ones that we're kind of thinking of. So the first is, what if my application is rejected? Reality is, I can't think of an application that has been rejected, and I think neither can my colleagues. So this is very rare. And the reason for this is that we work along with you if you had interest in this data, to make sure that there's no overlap, for example, with existing projects. So usually projects, if they're unique and if data protection requirements are met, applications are generally approved. And if there is anything not okay, the base team collaborates with each applicant to kind of refine the submission to ensure that it can be approved. How long does this approval process take? It's relatively fast. So of course you need to prepare what you are interested in and kind of get this together. But once this is done and it's submitted, the base steering committee decides on each occasion. So usually 2 to 3 weeks, plus or minus. Also, are there specific deadlines when you can submit these applications? That's not the case. This can happen continuously, and we decide on the data usage also continuously. So can you apply for all the data? Let's say you are interested in a specific question, but you're not so sure yet. So you know that we assess medical data. Can you just ask for all of it and then later decide? No, you shouldn't. And the reason for this is that part of this application process is also to have a detailed documentation of which projects are ongoing, and to ensure that there's no overlap. So usually we provide you with the data that you have requested specifically for your research project. That being said, it of course does not mean that once you have asked for specific data, you can't ask for other variables. Like, we're fully aware that research changes, you have to do a revision, you have a different question, then other variables can of course be also assessed. I don't know if this is the case here. I'll still say it because sometimes it happens. You have old data, you have used base data before. Can you just reuse it and add 2 or 3 variables and you don't have to write an application again? No, you shouldn't. Again, the same reason. We want this also for documentation purposes. So each project should have a submitted application. Can you share the data with others? I mean, generally speaking, everybody who is part of the data application can use the data, but it should not be shared outside of this scope or outside of the data protection agreement. One of the questions that were asked, but I think that is clear already, this is a study that has been going on for over a decade. Is the data already been used externally, and if so, how? Yeah, we have hundreds of applications that have been approved and have also been published. And you find the list, as said before, on the website where you can see all of the publications that have been using base data. Dennis, if you could go to the next slide. Can I collaborate with other researchers on my project? Of course, just because we have this data application process, you can collaborate with whoever you want to collaborate, as long as you put the name on the collaboration. There is nothing that speaks against that. And then also from a very practical perspective, an important point is, as you've seen, this is a 10-year longitudinal study, which is not only multi-institutional, but then we have different sub-studies. So it's quite a complex design in that sense. And it is possible that maybe on the first go, you're not aware of what is there, if all the variables are there that you need, if you want to have them longitudinally, if you want to have them cross-sectionally. And so the base team works very closely with each of the collaborators to ensure that you identify the variables that meet your research needs. So even if you're interested in the study, but you're not fully sure yet what to use, just write us, contact us, and then we will help you along the way. Can I publish the raw data for replication purposes, open science? So the base data itself cannot be publicly shared, which is obviously not a surprise, but of course you can publish the code and you can publish other things that are aligned with the data protection rules that we have in line. And then last but not least, can I use all the variables, or are there certain restrictions that some variables are only used internally and others maybe are used externally? And here, in general, all data can be used and you can apply for all the data that we have. However, that being said, they're highly sensitive data that we have that even we cannot use and put on our laptop. This is, for example, georeference data or other data. So this data then cannot be shared directly, but we have established a procedure through which this data can still be used in collaboration with our members. Yeah, and I think that's that for data usage. But of course, if you have any other questions, feel free to ask.